Today we'll explore sexual reproduction in flowering plants, focusing on the structures and processes you need to understand for class 12 and NEET. We'll move from the big picture of flower organization right down to the cells and events that give rise to a new plant. Begin with the reproductive architecture of a flower. The male and female organs are arranged to maximize successful fertilization. The stamen, made up of anther and filament, produces pollen, while the pistil or carpel, with its stigma, style, and ovary, receives pollen and houses the ovules. Petals and the receptacle help position and protect these reproductive parts. If we look inside the anther, we find a layered structure that supports pollen formation and release. From the outer epidermis through the endothecium and middle layers to the nutritive tapetum, each layer has a role. The tapetum nourishes developing pollen, the sporogenous tissue gives rise to microspore mother cells, and dehiscence of the anther ultimately releases mature pollen grains. Microsporogenesis begins when a microspore mother cell undergoes meiosis to produce a tetrad of microspores. Those microspores then differentiate. Vacuoles form, nuclei reposition, and an asymmetric mitotic division produces a large vegetative cell and a smaller generative cell, the first step toward creating the male gametophyte. The mature pollen grain or male gametophyte is a compact unit built for delivery. It has an outer exene and inner intine with a germ pore for tube emergence. Inside, the vegetative cell contains the vegetative nucleus that controls pollen tube growth, while the generative cell or its nucleus will form the two sperm cells needed for fertilization. On the female side, the anatropous ovule shows key features. The integuments form the future seed coat, the nucellus nourishes the embryo sac, and the chalaza marks the basal end, while the micropyle provides the entry point for the pollen tube. The orientation and protective layers of the ovule are important for successful fertilization and seed development. Megasporogenesis mirrors the male pathway, but with different outcomes. A megaspore mother cell in the nucellus undergoes meiosis to produce a dyad and then a tetrad of megaspores. Typically, only one of these megaspores remains functional while the others degenerate, and this functional megaspore will develop into the embryo sac. The mature embryo sac, or female gametophyte, is organized into specialized cells. At the micropylar end, you find the egg cell flanked by two synergids. The central cell carries two polar nuclei, and at the chalazal end lie three antipodals. The synergids have a filiform apparatus that helps guide the incoming pollen tube toward the egg. Pollination can be driven by abiotic agents like wind or by biotic agents such as insects, and each route brings characteristic adaptations. Wind-pollinated plants expose their stamens and stigmas and produce abundant lightweight pollen, while insect-pollinated flowers are often colorful, scented, and nectar-rich to attract bees, butterflies, or other pollinators. Once a compatible pollen grain lands on the stigma, it hydrates and germinates, sending a pollen tube down through the style. That tube navigates toward the ovule, guided by signals from the synergids, and the two male gametes travel inside the tube toward the embryo sac to accomplish fertilization. Double fertilization involves two distinct fusion events. One male gamete fuses with the egg cell to form the diploid zygote. This is syngamy, while the other male gamete fuses with the two polar nuclei to form the triploid primary endosperm nucleus, a process called triple fusion. The resulting zygote and endosperm begin the coordinated development of embryo and nutritive tissue. Embryo development then follows characteristic patterns in dicots and monocots. Dicots typically form two cotyledons along with a plumule, hypocotyl, and radicle, while monocots such as grasses develop structures like the scutellum, coleoptile, and coleorhiza that protect and support the young shoot and root during germination. Finally, the seed and fruit preserve the next generation. The seed coat derived from integuments protects the embryo and its food reserves. Cotyledons or structures like the scutellum store nutrients, and features such as the micropyle remain as functional openings. The surrounding fruit tissue, often arising from the thalamus or ovary wall, aids protection and dispersal so the cycle can begin again. That's it for today. Learn smarter with EduRev. Notes, test, and practice in one app. Download EduRev now on Play Store and iOS App Store